Okay, here we are, and now we're finally going to get into a little detail about mitosis. And once again, I'm going to preface this a little bit by saying, actually, there's a lot more detail than I'm going to get into, but we're going to break down my little acronym PMAT, the Prophase, Metaphase, Anaphase, Telophase. And before we go there, backing up just a little bit, we're going to look at that chromosome structure again a little bit more detailed. So chromosomes contain both the DNA that we're always talking about, that's going to hold all the information for making proteins, all the information about heredity, and a whole bunch of proteins. And so collectively, proteins are known as chromatin. So chromosomes have both the DNA and these proteins known as chromatin. And chromosomes that are dividing are made up of two identical pairs of sister chromatids. So once we've gone past that first restriction point, we know we're going to divide, and we've actually replicated our DNA. Once again, if our DNA is all condensed pre-mitosis, it would look like the image on the right, where we have two pairs of sister chromatids held together at a centromere. So once again, we have our sister chromatids, and this is a duplicated chromosome. Notice it's still one chromosome, and this is what I pointed out earlier in the very first lecture. Over here is just also one chromosome. It's just this one chromosome has replicated in that S phase, right, that we looked at, that S phase of interphase, the S stage, I should say. So it's replicated itself. It's still one chromosome that has double the DNA, but it's one chromosome. So this sister chromatid over here is identical to this one over here. And likewise, this sister chromatid over here is identical to this sister chromatid over here, all held together at this central region called the centromere. And once again, I mentioned earlier, for me this was a little confusing. Like, wait a minute, this thing is twice the size. Shouldn't we count that twice? No, this is still one chromosome. It's doubled its DNA in that S stage of cytokinesis. It's preparing for separation within mitosis. Now we're showing it to you in this way, but remember, most of the time the DNA is not all coiled up very pretty like this. Normally it's all a little bit more unwound within the cell, and it only coils up, actually we're going to see right now, in mitosis. So a couple other organelles that are going to be very important, other cell structures that are going to be important in mitosis. So we're just talking about some structural components right now before we jump into it. We have what's known as the centrosome, as opposed to, once again, the centromere we just talked about. The centrosome is the microtubule. So remember, when we think microtubule, we think of specialized proteins, okay, organizing center of the cell. So these specialized proteins that are usually structural for like cytoskeleton, movement, those type of things. And that's important, that concept of movement here, cellular movement within the cell. So the centrosome is the organizing center, so just an area. And right here we have two. Okay, we have over here, this is the centrosome area, and over here, the centrosome area. Then we have what's called asters. So this is an array of those microtubules at the ends of the cells, what's known as the poles. So what's going to happen is that the centrosomes, the area, the center of the cell, is going to be at the opposite poles. It's going to send out asters going out like this. So think about, once again, like a little infrastructure on all directions, kind of bracing itself within the cell. And specifically, we're going to have these microtubule spindle fibers that are actually going to connect and line up those chromosomes smack down in the middle of the cell. All right? And within that area, that centrosome, that microtubule organizing center, we have these things called centrioles, which are cylinder-like structures. Okay, you can see they, and they arrange at right angles to each other. And this is what's actually going to assist in forming these spindle fibers that are going to go out, these proteins that are going to line up the DNA smack dab in the middle of the cell, getting it ready for replication. So really what we're talking about right now, once again, are just specialized components in the cell, primarily what, as always, is going to be specialized proteins that are going to line up that DNA in the middle of the cell and then pull apart the sister chromatids, is what we're going to find out happens in mitosis. So we have that separation for two daughter cells. So once again, here's our overview. You've already seen it. We have, once again, a parent cell up here. All right, beginning of interphase, G1. This is G1 here. Then we have the DNA replication. That's the S. And then after that, we have G2 phase. Now notice, do we increase our number of chromosomes? No. No. Do we increase our DNA? Yes. So the number of chromosomes we have here in this example, this is not the human, this is just an example here. We have the diploid number, the 2 in number, diploid. And for this particular organism, we're saying this organism has 4 chromosomes. Now, we come down here to the G2 phase, we still have the diploid number. 2 in, 4 chromosomes. 1, 2, 3, 4. We still have 4. But we've doubled our DNA in this S stage here. Then what's going to happen? We're going to line up these 4 chromosomes with our double DNA, and we're going to separate them apart. So once again, we come down here and we have 2 daughter cells that are genetically identical to this first one up here, and they are, once again, the diploid number, the 2 in cells. They still have 4 chromosomes. We never lost the 4 chromosome number. So a diploid cell makes and divides an exact copy in its nucleus. When is this used? Cell growth and repair. This happens trillions of times within your lifetime. Actually, way more than trillion, excuse me, I forget the exact number, but it's within a typical person's lifetime. I want to say it's the number of cell divisions through mitosis is astronomical. If I can find it before my next lecture, I'll find that exact number. But this occurs in body cells, the vast majority of cells in your body. And once again, there's my PMAT, my 4 phases of mitosis. Prophase, metaphase, anaphase, telophase. So let's just quickly look over each one of those. So, prophase, the first one. What's going to happen here, this first stage? Well, we've already, remember, gone through that S stage earlier in interphase. So we know we have that double the DNA. So they've already doubled up. But now what's going to happen is that the chromosomes are finally going to condense and become visible. Now remember, prior to this, prior to prophase, prior to mitosis starting, the DNA really isn't coiled up yet. They're kind of all over the place, uncoiled. But in early prophase, they start to condense and get this look that we're used to. Kind of looks like an X, right? What is also going to happen? Well, remember, we need to duplicate the DNA that's within the nucleus, but we also have to separate out the nucleus. So there's a lot of stuff that has to happen, and we have to line up this DNA. So what also is going to happen is this nuclear envelope, this nuclear membrane, this nuclear envelope is going to actually start to fragment and disappear. So we actually lose our nuclear envelope. And then our nucleolus disappears. And here's the important part here. The centrosomes, remember those we just talked about, are going to start to move to the opposite poles. So look at how we're moving to opposite ends over here. And you can actually really see it well over here. We can see those centrosomes on opposite sides with the duplicated chromosomes starting to line up in the middle. And you see those spindle fibers appear and attach to the centromere. So we have spindle fibers attaching to the centromere, and that's going to be those lined up in those areas, those centrosomes, moving to the opposite side. So right now this prophase, it's preliminary. Think of it that way. It's just getting things ready. Then we move to metaphase. Now, this is when we actually line up those chromosomes. The ultimate goal of metaphase is to get along the center line here, right down the center. And in fact, that center is called the metaphase plate, along the equator of the cell here. And at this point, by the time we get to the end of metaphase and we have our metaphase plate, once again, you can see it really well up here. We can see these spindle fibers coming off up here. You can actually see it in this micrograph here. We have that nice metaphase plate going down the middle. That's how we know we're done with metaphase. We've lined everything up, getting ready to divide them apart. Then we get to anaphase. Now, this is when we really start pulling apart. Now, remember that they're all lined up down the middle initially in metaphase, and yet those spindle fibers that actually attach, here's the spindle fiber on each side from the centrosomes attaching here at those centromeres. Well, now what's going to happen is those spindle fibers are going to pull them apart. They're going to pull them apart so sister chromatids separate and become daughter chromosomes. So at this point in time, technically, if we really wanted to count it up, we'd actually have even more than the diploid. We don't really look at it from that standpoint, but we would have, in this example, we'd go from 4 to 8 at this brief moment in time. Once again, we don't technically look at it that way, but this way, this is what's important. Each daughter cell is going to get an equivalent amount of the same chromosomes, separating the duplication. So in a way, the analogy is in interphase, in that S stage, we've made a copy. Okay, we've made a copy, like a photocopy of a document. Right? We made a copy of this document. I'll put I got a document. I'm going to photocopy it. I'm going to make another copy of it. But you know what? I've stapled it together. I've stapled them. Maybe not the best analogy, but hopefully this works. And then finally, when we get to mitosis, we line up the copies in the middle and we just pull apart from the staples, and they go into 2 different stacks. The papers go into 2 different stacks. We break the staple. So now I've got my 2 different copies in my 2 different piles, my 2 new cells. Finally, we have telophase or telophase, depending on how you like to pronounce it and who the teacher is. You can pronounce it either way. I always say telophase, but that's how I was taught with my professors. But the animations always say telophase. And then cytokinesis. So once again, telophase is the end technically of mitosis, and cytokinesis is different. Mitosis is specifically referring to separating the DNA and creating 2 new nuclei, if you will. Cytokinesis is separating everything else. Well, once again, these 2 overlap as far as what's really happening here. So what's happening? The chromosomes arrive at the poles. They become indistinct in chromatin again, which means they no longer look nice and pretty like this. They unwind and start looking more like this again, which is how they really look for the vast majority of their life. The nucleoli reappear in each individual nucleus. The spindle disappears. The nuclear envelope reassembles, and guess what happens? Here's the key. 2 daughter cells are formed. and by a ring of actin and a cleavage furrow. In other words, we've now created two new daughter cells, and this is where it happens. In fact, once again, I've already shown you this next animation, but I want to show it to you again. I know you've already seen it, but now look at it. Now that we've gone over a little more of the detail, look at it again as far as the steps within mitosis and cytokinesis.
Mitosis is a process of nuclear division by which replicated copies of a cell's DNA are organized into chromosomes. The identical copies of the DNA are then divided equally between two daughter cells. Early in mitosis, during prophase, the nuclear membrane and nucleolus begin to break down. As prophase continues, the DNA condenses to form chromosomes, while the mitotic spindle, made of proteins called microtubules, begins to form and attaches to the kinetochores on each chromosome. During metaphase, the chromosomes are moved to the central plate of the cell by the spindle microtubules. In anaphase, the microtubules attached to the kinetochores of each chromosome shorten, drawing the chromatids of each chromosome to opposite ends of the cell, while the unattached microtubules elongate, stretching the cell. During telophase, the chromatids, now called chromosomes, reach the poles of the cell as the unattached microtubules continue to elongate. The nuclear membranes begin to reform as the nucleoli reappear and the chromosomes relax as the DNA unwinds. Telophase is followed by cytokinesis, or division of the cytoplasm. In animal cells, a cleavage furrow forms at the center on either side of the cell, pinching through until the original cell is divided into two daughter cells. In plant cells, cytokinesis is different due to the presence of a rigid cell wall.
So once again, there's the basic breakdown of mitosis and cytokinesis. And for right now, that is the depth I want you to know it in, the same depth as the book. I'm not going to go any deeper. Once again, in the future, a little bit more in anatomy and physiology and so forth, we might go a little bit deeper within mitosis and cytokinesis. But for right now, I just need you to get the concept of, okay, in normal growth of the body cells and the vast majority of cells in your body, this is how the cells duplicate. This is just basic cellular reproduction. And I need you to get the steps down of, okay, we've got the interphase, that G1, S, and G2 phases. And once again, there are these checkpoints along the line. And then mitosis and cytokinesis, PMAT, prophase, metaphase, anaphase, telophase. What happens in each of those steps and how we're actually going to be able to separate this DNA to ensure, here's the important part, that we have two identical daughter cells.